Welcome to the deep dive. Today, we're jumping into, well, a really exciting area, biotechnology and gene therapy.
Absolutely.
It feels like more than just science advancing. It's almost a revolution in how we can treat diseases, right? Tackling them at the source.
It really is. We're seeing decades of research finally turning into actual therapies.
Hmm.
So yeah, in this deep dive, we'll look at some of these breakthrough treatments, try to unravel how they actually work, the science bit.
And the journey they take, because it's not simple.
Not at all. That whole complex path to actually getting them to patients, we'll be drawing on quite a range of materials to piece that together.
Okay, so these amazing therapies, they don't just pop up fully formed. Where does that first glimmer of an idea come from? Is it like a lightning bolt moment or something else?
It's usually much more methodical than a lightning bolt, I'd say. It often starts by really understanding unmet medical needs, meaning looking at diseases where the current options just aren't good enough, or maybe there are no options at all. That deep understanding, plus what we're constantly learning about the biology of these diseases, that's what points towards potential ways to intervene.
Right. So identify a gap, a need, then dig into the biology, what's actually going wrong inside the body. And once you have a handle on that, you start looking for a place to step in.
Exactly that. Once you've got a clearer picture of the disease mechanism, the next really critical step is target selection.
Target selection.
Yeah, finding that specific molecule could be a protein, maybe an enzyme, sometimes even a gene that's playing a key role in driving the disease.
Yeah.
The aim is to find what we call a druggable target.
Druggable. Okay, so not just any molecule involved, but one we can actually affect with a drug. Like, it's not enough to know a wheel is broken. You need to find a wheel you can actually reach and fix.
Perfect analogy. And it's not just about finding a keyhole. You want the right keyhole. Sometimes you find a target that when you tweak it, it influences a whole network of disease pathways. That can be much more powerful.
Ah, like hitting a master switch instead of just one light bulb.
Precisely. And this ability to actually change the target's activity, we call it modulability. That's also key. You need like a lever you can actually pull or push to get the result you want.
Makes sense. So, okay, we found this promising lever. How do you know pulling it will actually help, that it'll have a therapeutic effect?
Good question. That's where target validation comes in. Researchers use different models, could be cells in a dish, could be quite sophisticated animal models, to basically simulate what happens when you hit that target.
Trying it out on a smaller scale first.
Exactly. It gives you that initial evidence. Does messing with this target actually do what we think it will? It's like running simulations before you commit to building the whole thing.
That sounds way more efficient. Sort of testing the blueprint before construction.
It is. And it's getting even more sophisticated now with big data and bioinformatics. We can analyze huge data sets, gene activity, protein interactions to help spot potential targets.
How does that work?
Well, you might look for genes that are always switched on too high in disease tissues, for example, or proteins that aren't interacting the way they should be. It's like using data mining to find the most likely suspects.
Okay, so we've used data, models, we've got a target, we think hitting it will work. Now the actual drug development part starts, right? Which sounds long and complicated. Where do you begin the testing?
All right, the development journey kicks off with preclinical evaluation. This is absolutely crucial. It's where we get the first real safety data before anything goes near a human.
Okay.
A huge part of this is toxicology studies in animals, testing for potential risks, side effects.
You mentioned relevant animal species before. Why relevant? Why not just say mice for everything?
Well, especially for biopharmaceuticals, things like antibodies or protein therapies. The international guidelines, like from ICH, say you need a relevant species. That means the animal's biology has to be similar enough to humans in the key area.
Meaning the drug actually works in that animal.
Yes, the animal has to have the biological target, the receptor, the molecule that the drug is designed to hit. If the drug can't interact with its target in that species, then testing it for safety might not tell you much about human risk.
Okay, that makes sense.
These studies also help check if the drug hits unintended targets in the body. Very important for predicting human safety.
So find an animal where the drug does its thing, see what happens good and bad. If that preclinical data looks okay, what's the next barrier?
Once you have enough preclinical safety data and hopefully some hint that it might actually work, the next big step is getting permission to test in humans.
The regulators step in.
Exactly. Yeah. In the U.S., you submit an investigational new drug application, an IND, to the FDA. Think of it as a big package of all your preclinical findings and your detailed plan for the human trials.
The protocol.
right? The clinical trial protocol. In the EU, it's a similar thing called a clinical trial application, or CTA. There's a huge amount of ethical review and regulatory scrutiny before any human testing can start.
So the FDA and others are gatekeepers right from the start. Okay, you get the green light, clinical trials begin. Can you give us the quick flyby of the phases?
Sure, it typically starts with phase 1, small group, often healthy volunteers. The main goal here is safety. What dose is safe? How does the body handle the drug? Absorption, metabolism, excretion, all that.
Just basic safety and processing.
Yeah. Then phase 2, now you're usually testing in patients who actually have the disease. Still looking closely at safety, but also starting to get the first signals of efficacy. Does it seem to be working?
Okay, first hint of whether it helps.
Yeah.
Then phase 3, that's the big one, right?
That's the big one. Phase 3 trials are much larger, involving potentially hundreds or thousands of patients with the disease, often across many different locations.
Why so big?
Well, the main goals are to really confirm if the drug is effective compared to maybe a placebo or the current standard treatment, and to keep monitoring safety in a larger, more diverse group of people. You need that diversity to see how it works in different subgroups.
Makes sense. You want to know it works for, well, everyone who might eventually take it. How do they measure if it's working in phase 3? What are they looking for?
They use predefined endpoints. These are specific things they measure to see if the treatment worked. There's usually a primary endpoint, the main outcome the study is designed to assess. Could be survival, could be reducing tumor size, could be improving a specific symptom score.
Something concrete.
Exactly. Measurable. Then there might be secondary endpoints looking at other benefits or aspects. You need solid data showing a real clinical benefit.
And you mentioned comparing it often to the existing treatment.
Yes, very often. Many trials aim for superiority, showing the new drug is definitely better than what's currently used, or better than nothing if there's no treatment.
Better is good.
Better is good. But sometimes the goal is non-inferiority. This means showing the new drug is at least as good as the current standard. That might be valuable if the new drug is, say, easier to take or has fewer side effects.
Okay, so maybe not a home run, but still a valuable improvement in some way. These trials must be incredibly carefully planned.
Oh, absolutely. The clinical trial protocol is the rule book. It details everything. Who gets into the study, how the drug is given, how data is collected, how it's analyzed. Everything has to be standardized to minimize bias and make sure the results are trustworthy.
And how they analyze the data matters too. You mentioned ITT and per protocol.
Right. Intention to treat, or ITT analysis, includes everyone who was randomized into the trial, even if they dropped out or didn't follow the instructions perfectly.
Yeah.
It gives a more real-world picture, maybe. Per protocol analysis only looks at the patients who stuck to the plan exactly. Comparing those results helps give a fuller picture of the drug's effects.
Sounds incredibly thorough. But we hear about phase 3 trials failing pretty often, right? It's not a guaranteed win, even at that stage.
That's absolutely true. It's a tough reality, a lot of investment, a lot of hope, but many phase 3 trials just don't meet their primary endpoint. The drug doesn't prove effective enough, or maybe safety issues emerge. And even for drugs that do get approved, sometimes problems crop up later after they're on the market. Ongoing monitoring is essential.
A sobering reminder. Okay, but let's say a drug makes it through phase 3, looks good. Then it's back to the FDA for the final decision.
Yes. The sponsor submits all the data, preclinical, clinical, manufacturing details, and what's called a new drug application, NDA, or biologics license application, BLA, for biologics. The FDA reviews everything meticulously. Safety, efficacy.
And the manufacturing part is key too.
Hugely important. They look very closely at the manufacturing process and the control strategy. How do you ensure every batch is the same quality, the same safety? This is where quality by design, or QBD, comes in.
QBD, like designing quality from the start.
Exactly. Instead of just testing quality at the end, you understand how different steps in manufacturing affect the final product. You control those critical steps to build quality and consistency into the process, like knowing exactly how oven temperature affects your cake, not just tasting it afterwards.
Got it. So the FDA is looking at the whole picture, product, and process.
Definitely. And they have real teeth.
Yeah.
They can put a clinical hold on a study if safety worries arise during trials. Sponsors also have to report serious side effects very quickly.
And the rules keep changing.
They do. Science evolves, technology evolves, so the regulatory standards adapt too. We see it with things like AI and medical devices now, and even which part of the FDA reviews what can shift. CBER has traditionally handled biologics, but some now go through CDER, the drug center. It's dynamic.
Definitely complex. Okay, let's shift focus a bit. Let's talk about some of the really exciting stuff, the breakthroughs themselves. What mechanisms are driving these new therapies? Any examples from the sources?
Oh, absolutely. There's some great examples. One is about using orally delivered siRNA, small interfering RNA.
RNA interference, right, silencing genes.
Exactly. This specific example targeted a gene called MAP4K4 inside macrophages, which are immune cells. MAP4K4 is involved in inflammation, so the idea is deliver this siRNA orally, it gets to the macrophages, silences that gene, and helps suppress systemic inflammation. Very targeted.
Orally delivered siRNA is pretty cool, much easier for patients.
Potentially, yes. Another fascinating one involves microRNAs, specifically microRNA 92 influencing something called KCC2 in certain brain cells, cerebellar granule neurons.
MicroRNAs are tiny regulators.
Yeah, they fine-tune gene expression. This just shows how we're drilling down to these really intricate molecular controls, even within specific cell types in the nervous system.
Amazing precision. What else?
Well, there's the development of HIV protease inhibitors, like nelfinavir and darunavir. That story really shows how understanding the exact shape and interactions between the drug and its target, the HIV protease enzyme, led to better drugs.
How so?
They figured out exactly how the drugs fit into a little pocket on the enzyme, the S2 pocket. Darunavir, for instance, was designed with a specific structure to fit really snugly in that pocket and make strong bonds, making it much harder for the virus to mutate and become resistant. It's like designing a key that fits even if the lock changes a bit.
Great analogy. And then there are the therapies that have really made headlines recently.
Absolutely. CAR T-cell therapy has to be mentioned. Chimeric antigen receptor T-cell therapy.
Taking a patient's own immune cells and reprogramming them.
Essentially, yes. You engineer their T-cells to recognize and attack their cancer cells. It turns the patient's own immune system into a targeted weapon. It's been truly revolutionary for some blood cancers, and regulators have definitely recognized it as a breakthrough.
And gene therapy, too.
Yes, like Luxturna. That's a gene therapy approved to treat a rare inherited eye disease that causes blindness. It delivers a working copy of the faulty gene directly into the eye. Another huge step forward, acknowledged by regulators.
CAR-T, gene therapy. It feels like we're really getting into futuristic medicine, but making these complex things must be incredibly challenging. Quality control sounds vital.
You hit the nail on the head. Manufacturing these advanced therapies is incredibly complex and needs extremely tight control. For many drugs, especially biologics, just controlling how the drug substance crystallizes is critical.
Crystallizes, like salt or sugar.
Sort of, but on a molecular level. How the molecules arrange themselves into a solid structure. This affects stability will it degrade, and bioavailability will the body absorb it properly. Things like polymorphism.
Different crystal forms.
Right, the same molecule packing in different ways. That plus particle size, purity, it all has to be controlled meticulously. Getting the crystal form wrong can actually lead to recalls. Regulators watch this very closely.
Wow, so the tiny physical structure makes a massive difference.
Massive. And even the inactive ingredients, the excipients, play a role. They can be used to help control the crystal size and shape, which then affects how the drug dissolves and gets absorbed.
Okay.
And then you need sophisticated tools to check you've made the right thing. Techniques like IR spectroscopy, infrared spectroscopy, act like a molecular fingerprint to confirm the structure is exactly correct.
So lots of checks and balances.
Constantly. And new manufacturing approaches are coming online too, like continuous manufacturing. Instead of making drugs in batches, it's a constant flow. That can potentially improve quality and safety.
Interesting.
And it all ties back to that quality by design idea we mentioned. Understanding the process, controlling it, building quality in from the start.
Right. It keeps coming back to ensuring every single dose is safe, effective, and exactly what it's supposed to be.
That's the goal, always. From that very first idea about an unmet need, through all the research, the trials, the regulatory hurdles, right down to the details of making it consistently. It's quite a journey for these biotech and gene therapies. It really showcases, scientific ingenuity, but also the absolute need for careful processes and oversight.
Well, this has been incredibly insightful, a real deep dive into biotechnology and gene therapy, definitely dynamic fields. We've gone from the spark of an idea through the maze of development and trials, looked at the regulators, and touched on some genuinely transformative therapies.
It's been great to talk through. These fields really do hold immense promise for diseases that seemed untouchable not long ago. But as we've seen, getting these breakthroughs from the lab bench to the patient's bedside is, it's a huge challenge. Complex, expensive, and with plenty of hurdles.
Yeah, it's clear that progress is constant, but the hard work in research, development, manufacturing it all has to continue to actually deliver on that promise and improve treatments for people. It really makes you think about what's next, doesn't it? And the importance of doing the science rigorously and ethically.
Absolutely. Hopefully this deep dive gives you a solid foundation. There's so much more to explore in specific areas if your curiosity's been sparked. It's a field that's constantly evolving.
Thanks for joining us on the deep dive.